Scaly fossil is the oldest known piece of skin. The specimen came from a 289 million-year-old fossil deposit and might offer clues to how skin evolved. Dry, scaly skin may be one of the least fun parts of winter. But in the broad scheme of things, a tough, watertight hide is part of what enabled the ancestors of modern reptiles, birds and mammals to move inland while their thin-skinned amphibian cousins remained close to water. In a study published Thursday in the journal Current Biology, scientists announced the discovery of the oldest known piece of fossilized skin. The pebbly scrap, which is no larger than a human fingernail, most likely belonged to an ancient reptile and provides rare insight into the evolution of skin. The piece of skin is one of countless traces of prehistoric life preserved in the Richard Spur limestone cave system near an oil seep in southwestern Oklahoma. When animals fell into the caves 289 million years ago, the conditions were ideal for preservation. Fine clay sediments rapidly buried the bodies, low levels of oxygen in the groundwater slowed the decay process, and hydrocarbons from the oil permeated the tissues and made them less hospitable to bacteria. The tar seeped into the fossils, staining them. In 2018, Bill May, a retired forensic analyst, shared some tiny flakes from the Richard Spur that he couldn't identify with Robert Rice, a paleontologist at the University of Toronto, Mississauga. "We got very excited by what we saw under the microscope," said Dr. Rice, an author of the paper. "The texture of the skin is quite unique and interesting. It really stands out from other fossil material. It's obviously not bone," said Ethan Mooney, a master's student who worked with Dr. Rice on the paper. If anything, the fossilized tissue bore a striking resemblance to the scaly skin of a crocodile. A PhD student and another author of the paper, T. Maho, used a diamond-tipped blade to separate a tiny portion of the skin into hair-thin layers. The outermost layers had hardened structures made of keratin, the protein found in mammalian hair and nails. These hardened structures, or cornifications, are a hallmark of the skin of amniotes, the land-dwelling, backboned animals including reptiles, birds and mammals. Amniotes, ancestors evolved to be able to live and reproducing outside of the water, unlike their amphibious relatives. Tough, impermeable skin was a key evolutionary adaptation for amniotes taking over the land, because in order to survive in terrestrial environments, you want to not dry out, Mr. Mooney said. The fossilized skin was found by itself, unattached to bone. However, Richard Spur has yielded countless fossils of a small, lizard-like reptile called Captorhinus agouti. While scientists haven't found a C. agouti fossil with skin attached to it, they did identify one with remnants of cornifications. Dr. Rice said that suggests the skin came from the same animal. Hans Sues, a paleontologist at the National Museum of Natural History who was not involved with the study, said that he was delighted by the paper, agreeing that it is really the earliest fossil example of skin. "We have had skin impressions, but here they can actually look at the detailed structure under the microscope as if it were skin they had just harvested from a living animal," Drive. Sues said. "And that's a really important discovery. Paleozoic cave system preserves oldest known evidence of amniote skin. Skin cast and compressions. A skin cast, ROMVP 88287, Figure 2a, and 7 compression fossils, ROMVP 88280, 88286, Figures 2d-2j, from the karst deposits of Richard Spur, have been recovered independently from one another and without association with skeletal remains. Although all skin patches display closely spaced, non-overlapping, and non-polarized tuberculate scales with a pebbled texture, there are significant differences in their size, distribution, and frequency. This extensive collection of skin patches likely represents various bodily regions across multiple individuals and possibly more than one species, while it is also possible that a few specimens may belong to a single species. The 3D skin cast is the first fossilized skin proper from the Paleozoic and displays convex hyporelief tubercles with the depths of the scales ranging between 25–35 micrometers, Figure 2a. The overall shape of the skin cast is convex with a few raised areas. A total of 85 irregularly shaped tubercles are on the two patches and have a frequency of about 13 to 16 per square millimeter. Thin, wrinkle-like ridges are observed below the level of the tubercles running parallel to each other, apparently connecting neighboring tubercles. Histological analysis reveals that the skin cast, figures 2b and 2c, is composed of an epidermis layer that is up to 50 micrometers in thickness and includes some corneous tissue, appearing as a lighter colored zone, 0.8 below the epidermis. There is a distinctive thin, dark, linear structure likely denoting the separation of the epidermis from the underlying dermis. Additionally, an identifiable hinge region is displayed between adjacent flat, epidermal scales. The preservational characteristics of this material, partly caused by hydrocarbon impregnation, is such that some of the finer integumentary structures are not identifiable, nor can detailed chemical analyses be undertaken. The compression fossils have a tuberculated surface with a slightly concave epirelief, figures 2d-2j. A wide range of tubercle sizes and shapes are displayed across and within individual patches. For example, RhomBP 88280, figure 2d, displays a scalation pattern transitioning from pebbly to polygonal. 0.19 tubercle sizes range from 60 to -540 micrometers in width when comparing all specimens. Overall, these surfaces appear as rounded tubercles that are densely arranged, as previously noted in embryonic sauropods. 0.202 of the compression fossil patches, RhomBP 88283 and RhomBP 88285, appear to preserve some curvature, figures 2G and 2I, while also having relatively smaller and higher densities of tubercles. Additionally, RhomBP 88285 has the highest density of tubercles, 40 to 66 per millimeter 2, and individual tubercles range from 60 to -171 micrometers in size, figure 2I. Interestingly, RhomBP 88281, figure 2e, had the largest tubercles with the lowest density, 9 to 10 per square millimeter. RhomBP 88286 has a unique appearance, displaying a glossy surface and sporting approximately 30 elongate tubercles, figure 2j, with a few smaller-sized circular tubercles that are irregularly dispersed. A few of the tubercles even appear to have longitudinal ridges on the central face, which would have been raised within the skin proper. Ossified integumentary tissues. Dermal scales. Ossified dermal scales are fairly common at Richard Spur, and we include here examples from terrestrial anemniote temnospondylus, lepospondylus, and other unknown tetrapod taxa, figure 3. Scales of the terrestrial temnospondyl predator Echoloma, figures 3c3e, are slender and spindle-shaped, with one end narrow and elongated while the other is expanded and rounded. A bulge-like crest is observed on one side of the scales, which is shown to be visible on the dorsal margin of the posterior edge of the gastral scales. 0.21 The majority of scales are arranged with the distal portion overlapping the proximal portion of another successive scale. This morphology and the arrangement is consistent with dermal scales likely widely distributed throughout various parts of the skeleton, including and extending beyond the ventral body surface. Unexpectedly, small spicule-like structures are found overlying the dermal scales, with some areas more densely packed than others, figure 3f. These may represent where hard tissues were embedded in the skin, but we are unable to make a definite determination. Dermal scale impressions are also preserved in articulated lepospondyle skeletons, figure S1. They appear as densely packed, parallel, rod-like units with some overlap, figure 3a. An overall net-like pattern of ornamentation is formed, which appears similar to other lepospondyle specimens. 0.22 Similarly, typical dermal scales that once covered the external body surface are shown overlaying the pterygoid bone of the palate of Lustrafuse, OMNH 73718. Occurrence of such scales on the palate is likely an artifact of preservation, figure 3b. The surface is ornamented with rod-shaped striae that run parallel to each other on a broad fan-shaped scale. A heavy ridge that appears to separate two sections is observed at the anterior edge of each dermal scale. Additionally, one segment has scales that run semi-parallel and appear to converge with each other. However, the actual convergence point is not visible. A similar dermal scale morphology and arrangement is noted for the anamniote tetrapods Microbrachus and Hyloplesian. 22. Two unknown tetrapod dermal scales are also represented, figures 3G3J, and these differ in appearance from the other dermal scales examined. ROMVP 88302 has non-overlapping long dermal scales that run parallel to one another, with an average width of 25.1 plus or minus 5.1 micrometers, figures 3G and 3H. There are several clusters of these scales that are noticeably curved and in various wavy orientations, some of which have a muted light green hue and others are more beige or brown. ROMVP 88303 has longitudinal scales that overlap with one another, with a slightly larger width of 43.8 plus or minus 6.8 micrometers, figures 3I and 3J. Unlike ROMVP 88302, the external surface of these dermal scales has spicule-like ornamentation covering one side entirely, while the other surface appears smooth. What are likely palpebral ossifications are also preserved in the orbital region of the stem-less amphibian Dolizerpodon, figure S4. These ossifications in the eyelid consist of a mosaic of ossicles that appear to be in almost perfect articulation, and they follow the edge of the inner orbit with a few also dispersed throughout the orbit. Although similar to other amphibamids 23 and those of micromelarpodontid dizerophoids like Micromelarpodon 24, these appear to be in better articulation with each other and conform to the spherical shape of the orbit. These ossifications differ from scleral ossifications, which form a ring of overlapping sub-rectangular plates in the eyeball and are also frequently preserved at Richard's Spur. 25. Discussion. Skin. The vertebrate fossil record is composed mainly of biomineralized tissues like ossified skeletal remains. The very rare occurrences of soft tissues and the associated preservational mechanisms are therefore of great interest. 26. Their low preservational potential is mainly attributed to the rapid action of decay microbes as skeletonization and fossilization through permineralization occur. 26, 27, 28, 29, 30 integumentary tissues of Mesozoic dinosaurs represent most of the relatively sparse occurrences of soft tissues in the terrestrial vertebrate fossil record. 13, 20, 26, 31, 32, 33, 34. However, little information is available for Paleozoic terrestrial vertebrates during their initial stages of diversification. 14, 35, 36, 37, when this outer layer of the body became a crucial protection and barrier, instrumental for successful terrestrial lifestyles. The surface morphologies of the skin impressions and cast are reminiscent of several extant and extinct reptilian integumentary morphologies. This pebbled, non-overlapping scalation pattern is similar to those of the extinct late Cretaceous hadrosaur dinosaur Edmontosaurus and extant crocodiles like Crocodylus. 38. 39. The hinge regions between epidermal scales and arrangement of the epidermis to the dermis also resemble those of limbless squamates, including the Amphisbaenian Diplomatopon Zerdni 40 and snakes. 16, 40. This is in contrast to the earliest evidence of an epidermis among the rhapsids, the lineage leading to mammals, that being impressions from the Middle Permian Esteminosicus and the Permo-Triassic Lystrosaurus, both exhibiting bulging ellipsoid tubercles with a tendency to partially protrude one another, producing an overall pustular surface texture. 41, 42. Overall, the observed morphologies presented here are consistent with variation in most extant and extinct reptiles, indicating that a non-overlapping, pebbled, tuberculated epidermis may have been the ancestral condition in amniotes, or at least in reptiles. The preservation of a terrestrial type of skin in an early amniote, with a general morphology similar to that of living reptiles, indicates that this important organ for terrestrial vertebrate life was already present at the initial stages of amniote diversification. In particular, the presence of an epidermis with hinges and scale-like protuberances underscores the importance of this component of the amniote skin as a barrier against the harsh terrestrial environment. We propose that this represents the ancestral condition for amniotes. Thus, It is the outer layer of the integument consisting of epidermal scales, representing the ancestral amniote state that was apparently involved in the evolution of avian feathers and mammalian hair follicles. 43 With the formation of complex follicles for feathers and hair through the invagination of the epidermis. Dermal ossifications. The presence of skin is probably the ancestral integumentary condition among tetrapods. 44 While external dermal scale covers are suggested to have originated even earlier among stem tetrapods, both being present prior to and within crown tetrapods. 45. 46. 47 The evolution of dermal scales during the Ordovician can be attributed to the ability of early aquatic vertebrates to biomineralize integumentary tissues to produce ossified integumentary structures. 46. 47. 48. 49. 50 Ossified dermal scales are therefore considered to represent the ancestral condition for tetrapods and are widely found among early aquatic and amphibious non-amniotic tetrapods. 21. 51 The fossil record has occasionally favored the preservation of ossified products of the dermis in early terrestrial tetrapods, but little information, rare impressions of the outer surface, is available about the dermis layer. Striking variation among dermal scale morphologies existed among early anamniote taxa during the early Permian at Richard's Spur, as seen in Figure 3. Some possess overlapping parallel arrangements of individual, long, rod-like scales, figures 3C and 3D, while others display gently curving, tightly packed, parallel scales, some of which do, figures 3I and 3J, and do not, figures 3A, 3B, 3G, and 3H, sport fine spicule-like ornamentations. Among living amphibians, only some members of Caecilia show any evidence of dermal scales, such as dermaphiids, ichthyophiids, and siphonopids, while others like scoliomorphids and typhlonectids have lost them, with the exception of Typhlonectes compressicata. 52 However, their morphologies do not appear consistent with those of early tetrapods, suggesting that they evolved independently, rather than representing the primitive condition among stem tetrapods. 50. 53 The ossified dermal scales of the temnospondyle Ascheloma are presented here, with the individual scales having a similar morphology to the gastral scales previously described. 21 For other aquatic and amphibious temnospondyls. Interestingly, the small spicule-like structures found overlaying the dermal scales may be carbonized remnants of the skin, but we are uncertain. Orbital ossifications, including palpebral ossicles, are also more common within the fossil record compared to skin, but rarely in the articulated manner shown here. What are likely palpebral ossicles of Dolizerpidon appear to have overlapping, rectangular plates articulated with one another, 23 protecting the eye, as in various extant lizards and crocodiles. 54. Taphonomy and hydrocarbons at Richard's Spur. The Richard's Spur locality in Oklahoma preserves the richest, most diverse assemblage of exclusively terrestrial vertebrates for the Paleozoic era. The terrestrial tetrapods, dating to the early Permian, around 289 to 286 Maya, are characterized by the impregnation of hundreds of thousands of fossil bones by oil seep-sourced hydrocarbons. 1 The significance of oil seeps is far-reaching, both in ancient 55, 56 and modern periods, often acting as natural animal traps such as the La Brea tar pits. 57, whereby organismal remains are infiltrated and encased by associated hydrocarbons. 58, 59, 60 The occurrence of well-preserved integumentary tissues, particularly skin, indicates that preservational conditions at Richard's Spur are exceptional among Paleozoic localities. The most common preservational mode at this locality is likely rapid allochthonous burial in fine clay sediments within the karst system, cave system formed by dissolution of surrounding limestone, figures S2a and S2b, during extreme rainstorm events, resulting in abundant disarticulated, and rarer articulated and partly articulated fossil remains. 4 However, there are other taphonomic processes, including the secondary reworking of fossil material within the cave system by groundwater. The extensive cave system at Richard's Spur is also characterized by successive fluctuations in groundwater levels, as well as oil seepage events that are recorded in the concentric growths of stalagmites and stalactites. 1 Figures S2c and S2d. The widespread presence of pyrite on external bone surfaces, within deep recesses of skulls, and even inside the bones, figures S3e and S3f, indicates an abundance of sulfur and anaerobic conditions prevailing in stagnant water within this cave system. This standing groundwater, rich in dissolved carbonate ions with largely anoxic or low oxygen environments, would have been particularly suitable for permineralization processes. 26.27 However, the interaction of ion-rich groundwater in conjunction with hydrocarbons is likely to have substantially increased the preservational potential of this environment for both biomineralized and soft tissues.